I'm going to talk about enzyme multiplied immunoassay technique and chemiluminescent immunoassay technique in this video. First, I will take up enzyme multiplied immunoassay technique. It does not require a solid surface to immobilize the antigen or antibody. Second, this technique is a non-classical competitive immunoassay. It is used for the detection of small molecules like hormones, drugs, drug metabolites in the body fluids like serum or plasma or urine. Here, it can be used for two purposes. One, for the clinical investigation to diagnose a particular disease, where sometimes the endocrine glands may be malfunctioning, therefore the hormone levels in the body may be less or excess. So that's how the hormone levels in the body fluids can be detected. Similarly, drugs and drug metabolites which are found in the body, which are used for the clinical purpose, if they are not acting in the person, it could reflect on the liver function. So therefore, the normal clinical investigation of these molecules by using this technique can help in diagnosis and pinpointing what is wrong in the person. Second, it can be also used for the forensic investigation, that is criminal investigation, where the hormones have been used for enhancing the performance by the sports persons. Similarly, the abusive drugs which are available in the market are used by the people, and under the influence of drugs, some criminal activities are carried out. At the same time, there are anabolic steroids which are also used for the performance enhancement by the sports person. These criminal investigation are carried out in suspicious cases by using the body fluid of the person, that is serum or plasma or urine, in which these components can be identified by using this technique, that is enzyme multiplied immunotechnique or enzyme multiplied immunoassay. Another advantage of this assay is that it can be directly investigated in the body fluid of the person, and there is no need of series of washing steps during the assay, which you have seen in other immunoassays, like whether it is radio immunoassay or ELISA. It is necessary that in each step of the assay, the sample needs to be given a thorough wash. In this case, there is no need of wash. Now, how this assay is carried out, by this diagrammatic representation, you will be able to make out. The principle involves the enzyme that is to be used in this assay is conjugated with the drug molecule. Now, this drug molecule, when I refer, it could be a hormone or the drug per se that is used for the treatment purpose, or it could be the hormones that are used by the sports personality for performance enhancement, as well as it could be even the abusive drugs. Like that, you need to prepare a model or the sample depending on what type of investigation you are going to carry out. So therefore, let us take a simple example. The drug molecule is conjugated to enzyme, and this enzyme, let us take a simple, that is alkaline phosphatase. Alkaline phosphatase is conjugated to the drug molecule, and the substrate for alkaline phosphatase is para-nitrophenol phosphate. So the para-nitrophenol phosphate, when it is reacted upon by the alkaline phosphatase enzyme, it will give rise to yellow color because of release of the phosphate molecule from the para-nitrophenol phosphate, which is in short form called PNPP. When the para-nitrophenol phosphate is conjugated or bonded with phosphate molecule, it is colorless. But on reacting with alkaline phosphatase enzyme, due to release of phosphate, it produces yellow color. Now, using this principle of the production of color by the enzyme substrate reaction, we move on further how the enzyme multiplied immunotechnique is carried out. Let us see the drug molecule conjugated to the enzyme is available with you, and you are investigating in the urine sample or serum or plasma sample of the subject the same drug molecule, or it could be the drug metabolite, or it could be an anabolic steroid or a normal hormone, whatever it may be. You need to have now readily that particular molecule available in the form of a complex with the enzyme with you. And you already have antibodies that are produced in the animals which are available in the market against all these drug molecules or drug metabolites or the hormones or the anabolic steroids. And these antibodies are specific for these drug molecules or hormones or the performance enhancing drug molecules. So the antibody can readily bind to the drug molecule through the FAB site. First, you take the sample to be investigated, and that sample, let us presume that it contains the large amount of the suspicious drug that you are looking for, and to that sample you add the antibody corresponding to that drug and incubate it for some time, mix it well, incubate it for some time. Later you add now the drug enzyme complex and once again incubate for some time, and then now visualize the reaction. Now the reaction will be in these two forms, either like if the sample under investigation contains large amount of the drug that you are looking for, and when you add the corresponding antibody pertaining to that drug into the sample, all the FAB sites of the antibody will be engaged by the drug. So now the antibody molecule has complexed with the drug molecule, and there will be no free antibody available now to combine with the drug enzyme complex that you add. Therefore, the reaction will be like in this slide, where the drug enzyme complex is capable of reacting with substrate and produce bright color. Same way, if the sample under investigation has drug molecule, it will have engaged all the FAB sites of the antibody. Therefore, drug enzyme complex is free now. Therefore, it can react with the substrate and produce the color. So this proves that the sample under investigation contains the target drug you are suspecting, which is being used as an abusive drug. The second possibility is that if the target sample does not contain the abusive drug or hormone or anabolic steroid that you are investigating, the antibody that you added corresponding to that particular drug will be engaged by the drug enzyme complex. All the FAB sites of the antibody will be engaged by drug enzyme complex. When the FAB sites of the antibody are engaged by this drug enzyme complex, it will not be available for now reacting with the substrate. The substrate para-nitrophenol phosphate that you added will remain as it is without getting split. Therefore, there will be no color. So this clearly indicates that the sample under investigation, if it produces the bright color, it is an indication that there is the drug in the sample. Therefore, it needs to be investigated further for the criminal investigation. But if the sample under investigation, if it does not contain the suspicious drug that you are looking for, then there will be no color formation as an end reaction. So this is as far as enzyme multiplied immunoassay technique concerned. It gets the name because large amount of enzyme will be available for the reaction, and it is like a multiple effect. Therefore, it is called as enzyme multiplied immunoassay. So we will now move on to the next immunoassay technique, that is chemiluminescent immunoassay technique. The name itself indicates that it is a chemical molecule which is luminescent in nature, and the chemical molecule which is luminescent in nature could emit light on its own, unlike in the previous case where the fluorescent molecule would require light to emit light at a different wavelength. In the luminescent molecule, there is no need of presence of light. It itself will emit light or photon at a particular wavelength. The molecule that is generally used for chemiluminescence assay is called as adamantyl dioxetane phosphate. So adamantyl dioxetane phosphate is a molecule which is inactive in the form of phosphate salt. But when the phosphate is cleaved from the adamantyl dioxetane, it will produce an unstable substance, that is adamantyl dioxetane, and phosphate will be released, and that adamantyl dioxetane will be luminescent in nature. For example, here the adamantyl dioxetane phosphate is going to be reacted upon by the enzyme alkaline phosphatase. It can cleave the phosphate from the adamantyl dioxetane. As soon as phosphate is cleaved from the adamantyl dioxetane, it will start luminescence. The principle of chemiluminescent assay is similar to the EMIT, where there is no need of multiple washings. It is a non-classical competitive assay, and it can be used for the investigation of the normal hormones, drugs, drug metabolites in the clinical investigation, as well as it can be used for the criminal investigation of hormones, drugs, abusive drugs, drug metabolites, and the anabolic steroids in the body fluids of the person. For this assay, once again, similar to EMIT, the drug molecule is complexed with the enzyme. As I said, the luminescent substrate is adamantyl dioxetane phosphate. Therefore, the enzyme to be used here is alkaline phosphatase. Alkaline phosphatase is conjugated with the drug molecule. That drug molecule you are going to investigate in the sample. Similar to the previous reaction, the drug molecule complexed with the enzyme can easily react with the substrate, and on reacting with the substrate adamantyl dioxetane, alkaline phosphatase will cleave the phosphate group, With the result, adamantyl dioxetane becomes an unstable substance and emits luminescence. Now let us see how the clinical or the criminal investigation is done in the sample. Similar to emit, the antibody corresponding to the particular drug to be investigated. When I refer here drug, it could be hormone also, or anabolic steroid also, or drug per se, or drug metabolite. So the antibody is added to the sample and incubated for some time, and after that, after the incubation, the drug enzyme complex is added. Like in the previous case, if the antibody molecules FAB sites, that is antigen binding sites, are all occupied by the target drug which you are looking for in the sample, the antibody will not be available to bind to enzyme drug complex. So when the antibody is not available to bind to enzyme drug complex, the enzyme drug complex being free can easily react with the substrate, and the substrate is adamantyl dioxetane, and the luminescence will be very bright. As soon as adamantyl dioxetane reacts with alkaline phosphatase, the phosphate group will be cleaved and you will get bright luminescence. And the luminescence can be visualized through luminometer, which will indicate the intensity of the luminescence. On the other hand, if the sample does not contain the target drug molecule you are investigating, it could be hormone, anabolic steroid, abusive drug, or abusive drug metabolites. If they are not present and a corresponding antibody is added, that antibody will readily engage the drug enzyme complex on its FAB site. With the result, the FAB sites of the antibody molecules will be occupied by the drug enzyme complex, and it will not be available to react with adamantyl dioxetane phosphate. Therefore, there will be no luminescence. Bright luminescence of the sample indicates the presence of abusive drugs or hormones or anabolic steroids under investigation, and no luminescence indicates that the sample is free of the drugs or abusive drugs, drug metabolites, hormones, and anabolic steroids. So this is how the investigation is done, and this is also a very simple investigation carried out in the solution form without multiple washings.